Hey biology students, it's Professor Gideon, and in this video we're going to tackle leukocytes and lymphocytes. And I've also written here that this would be a lecture to view before you do Lab 26 on white blood cell morphology. This lab is going to ask you questions specific to these white blood cells. And usually what we do in that lab, you view microscope slides of blood smears and you look for particular characteristic white blood cells. So we're going to point that out as we go through this particular lecture. So first of all, recall that all blood cells originate in the bone marrow, in the red marrow, and in the red marrow, we actually have the origin of several different blood cells. So blood cells originate in the bone marrow. And we're talking about red blood cells called erythrocytes, RBC, red blood cells, as well as platelets. Those are your blood clotting cells, and then leukocytes and lymphocytes, which we call white blood cells. Okay, so we're going to focus in on the origins of the leukocytes and the lymphocytes. So let's take a look at bone. Lovely there. Okay, so we're talking about the bone marrow. And in the bone marrow, we have stem cells. And a lot of times we use this term pluripotent to refer to these stem cells. And what that word pluripotent means is that they're undifferentiated, meaning that these are cells that haven't decided what type of blood cell they're going to differentiate into. Excuse me. Okay, so two particular types of stem cells. So this pluripotent stem cell differentiates into either what we call myeloid stem cells or lymphoid stem cells. And so these different further differentiated stem cells give rise to specific groups of leukocytes and lymphocytes. So the myeloid stem cells give rise to neutrophils, eosinophils, basophils, mast cells, monocytes, which further differentiate into macrophages, and dendritic cells. Okay, so that's the rundown of the myeloid stem cells. And then we're going to go through, we'll talk about each one of them. We're going to talk about what they look like under the microscope. We're going to talk about their job or their function in the immune system. These are the players of the immune system. They play an important role in the story of how this is a coordinated defense against the pathogen. And if you recall, all of these white blood cells that I've listed so far, all differentiating from the myeloid stem cells in the bone marrow, okay, all 6 of these here on the left side, these are all part of, if you recall, second line of defense. So in the last video, we described the overview of the immune system. We described how there's 3 lines of defense against pathogens, where the first line of defense primarily is physical, meaning anatomical barriers like skin and cilia and mucus, as well as chemical barriers like lysozyme and stomach acid, those sorts of things. And then we have second line of defense coordinated by leukocytes. So these would all be described as leukocytes. Okay, or white blood cells, players in the second line of defense. Do you remember if second line of defense is a component of innate or nonspecific immunity, or is it the adaptive/specific immunity response? That's right, it's nonspecific or innate immunity. So this is a component of innate immunity, which is also referred to as nonspecific, which means that it's the same response regardless of the type of pathogen. So it's not a specific, it's a one-size-fits-all response. These cells will pretty much act the same regardless of the type of pathogen that invades. They each have their specific job in the immune response. So technically there's actually one more that's part of second line of defense, and it is differentiating from the lymphoid stem cells. So lymphoid stem cells, we consider these to be specialized, the cells that differentiate from lymphoid stem cells to be specialized leukocytes called lymphocytes. Okay, because they differentiate from a different type of stem cell than the other group. They differentiate from the lymphoid stem cells in the bone marrow. So one more that falls into this category of second line of defense, and these are called natural killer cells. That's a pretty cool word, actually, natural killer, like born to kill. Okay, sometimes they're also referred to as NK, capital N, capital K cells. These are also part of second line of defense. So let's draw an arrow connecting them here. Also part of second line of defense in coordination with these other 6 leukocytes that differentiate from myeloid stem cells. So they're also innate immunity or one-size-fits-all response to an invasion. So there's 2 more groups of lymphocytes rather. So natural killer cells, and then we have T cells and B cells. And if you recall, T cells and B cells are the players in the third line of defense of the immune system, which is also referred to as adaptive immunity or specific immunity. Because these cells coordinate a very targeted approach to the pathogen, they are specifically chosen and carry out a response to target exactly what pathogen has invaded your body. And more on that later. So now we're going to go through each of these types of leukocytes. Let's go to the next slide. So this is starting with neutrophils. So this is a neutrophil. And what we're looking at here is how, in the next series of slides, we're going to see these stained leukocytes. And we call this process of staining leukocytes, we call that histology. And you have a feel for this now because we've been doing staining of prokaryotic cells. We've been staining bacteria using various dyes and the gram stain, and the spore stain, and the acid-fast stain. You get a feel there that we could stain cells with different colored dyes, and some cells will pick up different colors in the staining procedure, and then they have this characteristic color to them that we can use to differentiate them. So we want to be aware of how we would differentiate various leukocytes if seen in a histological stain. So this is actually showing us a neutrophil in a blood, what we would call a blood smear. So if we took a blood sample from somebody and we looked at what blood cells we would see in a blood smear, primarily what you would see actually would be all these pink circles. You see all these pink circles up here and here. These are all red blood cells, RBC, red blood cells. And so primarily that's what our blood is composed of, red blood cells. But occasionally you would see these white blood cells, and they're going to be stained mostly purple, pink. These are the dyes that we use in the histological stains. So we want to be aware of how they appear. So if we look at somebody's blood smear, we would expect neutrophils to have a relative abundance. And here we're going to look at percent of total blood cells, what the normal range would be. And this is 60 to 70% neutrophil. One of the characteristic features of seeing neutrophils in a blood smear is that if we look at their morphology, if you remember that word, just refers to their shape. So neutrophils have this very characteristic multi-lobed, and a lot of these cells actually have this as well, multi-lobed nuclei that stains purple. So when we see it, we see all these multiple lobes to the nuclei, okay? And that would be stained purple. Okay? And the cytoplasm. So the cytoplasm of the neutrophils, well, the cytoplasm usually stains pink, although it's also possible for it to be staining purple as well. Stains pink or purple. Okay. How about the job of this particular leukocyte, the function. So the function of the neutrophil is phagocytosis. And in I believe the next video that I'll record, we'll talk about the process of phagocytosis. And this word, it actually comes from the Greek word to eat. So phagophago means to eat. And cytosis refers to cell engulfment. Okay, so this gives you an idea of what neutrophils are going to do to invading microbes. They're going to eat them. In a nutshell, that's what's going to happen. There's a step-by-step process to that and some important things you should know about phagocytosis, but we'll leave that to the next video. Neutrophils oftentimes are the first phagocytes to the site of an infection. The site of infection. Now, be thinking about how we could use a laboratory diagnostic test to learn about what's happening inside of our patients' bodies. So a lot of times doctors will order a laboratory diagnostic test. Basically, you're gonna go in and you're gonna go see the phlebotomist and they're gonna take a blood sample. And what they're gonna do is they're gonna do something called a CBC, which stands for complete blood count. What they're going to look at is the abundance of these various leukocytes, as well as other blood cells. They can look at all of them, but they're going to look at the white blood cells in the event of an infection. And so you can imagine that if your patient had an active infection somewhere in the body and you took a blood sample, this 60 to 70%, which represents normal range in the body, if that is elevated, so if it's elevated to be greater than that number, which already is a pretty high number because there's a lot of these guys just in circulation throughout your blood, just waiting for infections and clearing infections constantly. That's their job. But if the neutrophils go really high, that means the white blood cells are having trouble clearing an infection, and that would be of interest to note in a CBC laboratory diagnostic test. Okay. Neutrophils are not the only phagocytes, by the way. So let's take note of who the other phagocytes are as well as we get through this slideshow. Okay, let's talk about the next white blood cell or leukocyte in our slideshow. So this leukocyte is called a basophil. And there's actually a related white blood cell, a leukocyte that's related to basophils, and it's called a mast cell. Now, both of these types of white blood cells have the same function, which is same function, which is to release histamine. So when you look at the microscopic view of the basophil in this particular blood smear, Do you notice it has all of these purple granules throughout the cytoplasm? So we call these granules, and you can view these in the histological stain. And these granules are actually full of histamine. Now, I'd like to mention at this point that something we're going to notice show up again and again in our discussion of the immune system is that there are these chemical signals that are very important to white blood cell communication and function. And so histamine represents one example of chemical signaling molecules, called cytokines. So cytokines, that's a very broad term to actually use in immunology, and it refers to a class of several different types of chemical signals that are secreted by leukocytes. So we'll add that, secreted by leukocytes. So now we've met one so far. We'll talk about more as we go, but one is histamine. Now, I bet a lot of you have actually heard of histamine, mostly in the context of allergies. So if somebody has an allergy to pollen or to cat dander, dog fur, dust, whatever, they might take an antihistamine medication to block the effects of histamine. Because what happens if you do have an allergy response to cats or dogs or certain types of foods? Well, what happens is you get inflammation, watery eyes, a runny nose. Those are all symptoms of a process called inflammation. So let's write here that histamine is released by basophils, and this triggers inflammation. And the symptoms of inflammation are oftentimes redness, swelling, increased fluid to a particular area. Now, all of this actually has a beneficial purpose, and I know inflammation is usually a negative connotation, but there is a purpose to it. It's a lot like fever. Fever also has a purpose in our immune response. So more on that though, we're going to do a video on inflammation. For now, just know that basophils, they release these granules full of histamine. And when they release histamine, we also have a word for that. We call that degranulation. So if a basophil degranulates, that means it releases histamine. By the way, mast cells, we're saying, do the same thing. So mast cells look very similar, and mast cells also degranulate to release histamine and trigger the inflammatory response of the second line of defense. The big difference, okay? Basophils are located in the blood. In this particular picture, we're seeing a basophil. Okay, this is a basophil. It's located in the blood, circulates via the blood vessels. And the relative abundance, we'll say relative abundance. So the normal range is between 0.5 to 1% of total blood cells. People who have allergies, if you did a CBC, a laboratory test, and you suspect allergies, what do you suspect you would find in their basophil count? That's right, their basophil levels would be high. So this is normal range again. This would be normal range. So elevated levels would indicate allergies. And a lot of that is actually genetic, and a lot of it is unknown. Why some people have allergies and other people do not. There's a school of thought in microbiology, I guess rather immunology, that it has to do with how you were raised as a child, whether or not you came in contact with dirt and dust and dander, that historically people used to be more exposed to those sorts of things. And that trains the immune system to recognize things like dust and dander and pollen and not initiate an inflammatory response where the body, essentially, if you're allergic to something, the body thinks that that's something, whatever that is, cat dander or whatever, is a pathogen. That's what the body thinks. And the body mounts an immune response against that particular allergen. And that's why people have these symptoms of inflammation. It's actually not a normal thing. And people can have really severe allergic responses where they have to avoid houses where people have cats or never take certain antibiotics like penicillin can cause an allergic response in some people. In the last video, we talked about bee stings initiating a very powerful allergic response that can lead to shock and can kill you. So anyways, a lot to talk about regarding allergies, but for now, just know that it is triggered by basophils when those basophils become activated and release histamine. And of course, antihistamine medications do work for a lot of people. So antihistamine medications block the effects of histamine and prevent the inflammatory response. So mast cells. Okay, so mast cells do basically everything that we just described regarding the basophils. The only difference, though, is that these are located in the tissues, not in the blood. So they're located in tissues, like the skin, okay, the epidermis. They're located in tissues, not the blood. But they have the same function. So if you get tissue damage, so you have a cut in your skin, and then suddenly it gets inflamed, with redness and swelling and pus and all of that. Well, that's thanks to a chain reaction event triggered by the mast cells that are located in your tissues, and they become alerted by tissue damage that there's a breach in the first line of defense, the skin barrier, and they're going to start degranulating and initiate inflammation. All right, so that's all I wanted to say about those. Oh, actually, you could see that here pictured, but one of the ways you can identify a basophil in a blood smear is that it has very characteristic purple granules and lots of them. Lots of purple colored granules. So you see just a ton in there. It also has there's a bilobed purple nuclei as well. But usually the abundance of the histamine granules take over the cytoplasm, and that's pretty much what you see. It's a very characteristic way to diagnose basophils. Okay, next. number 4. Okay, these are eosinophils. And so eosinophils, their relative abundance in the blood is 2 to 4%, normal range. So one of the things you notice about the histology of the eosinophil is that it does have a large bilobed, so it has a bilobed purple nuclei, a lot like everybody we've seen so far. So what's different? Well, eosinophils also have a cytoplasm chock full of granules, but these are a different type of granules. And these granules stain pink, different than basophils, where the granules stain purple. And there's lots pink granules. So we just see tons and tons and tons of them all throughout the cytoplasm. That's going to be your clue that we're looking at an eosinophil. So eosinophils actually have a couple different jobs. They do degranulate, so it is possible for these granules to be released by eosinophils. So we'll write that over here as functions. So the main job of eosinophils would be to degranulate, to release granules, and these granules actually are toxic enzymes called granzymes. So they release these granzymes for what we call extracellular attack of parasites. What type of parasites? Well, usually parasitic worms is usually what we're talking about here. We haven't covered parasitic worms yet in the course, but parasitic worms can invade the blood, and I love this particular video clip of that. So here we see a parasitic worm, and we see all these eosinophils from the blood coating this worm, the surface of the worm. They're attracted to the worm and they coat it. And as they coat the worm, they degranulate and release their granzymes. And what they're doing is they're digesting the worm. So this worm is huge, so they can't really swallow it, but they can digest it extracellularly. Okay, so it's an extracellular attack on parasites. If the parasite is small enough for the eosinophil to phagocytose it, which means to eat it, then it will do so. So the second function, we say that it's somewhat phagocytic. It's not the main collection of phagocytes. Remember, who's the other phagocyte we've talked about? That's right, neutrophils. So it's somewhat phagocytic, which means it can perform phagocytosis, which is a method of intracellular digestion of pathogens. So if somebody has an elevated eosinophil count, what is that a clue of? That's right, probably a parasitic worm infection would be the clue. Okay, monocytes. I forget, is this number, this is, okay, white blood cell number 5. Okay, white blood cell number 5, monocytes. Okay, so monocytes are actually inactive. They patrol in the blood in their inactive form. And they represent, see, their relative abundance would be 1 to 6% of total blood cells. And they patrol in this inactive form. How do we know that we're looking at a monocyte? Well, it has a very large bilobed nucleus. It's pretty huge. It's purple, stained purple. Takes up most of the cytoplasm. And then the cytoplasm is usually stained either pink or sometimes a light purple. So what happens is if an infection or a pathogen is present, the monocyte will differentiate into a macrophage. So this is a macrophage down here. Sometimes you see this symbol M like that, meaning a macrophage. So the macrophage looks different than a monocyte. Do you notice that it has a circular nucleus? Circular dark nucleus? Now, macrophages are not in the blood. These are located in the tissues. So when a monocyte becomes activated and differentiates into a macrophage, then that macrophage will migrate out of the blood into the tissues to perform its job. And the function of the macrophage is phagocytosis, intracellular pathogen destruction. So it's a phagocytosis. It's actually king of the phagocytes, okay? Macrophage means big eater. It's a big eater in the process of phagocytosis. Although with phagocytosis and, let's add something here, antigen presentation, and we'll talk about that in the next video, about what that means. Notice that the macrophage has sort of these jagged edges too. That might be another clue that we're looking at a macrophage. Jagged edges. But once again, you wouldn't see macrophages in a blood smear. You would see it in tissue histology, which is possible to do. Take sections of tissue and look at that and stain it and look at it under the microscope. Okay, the last little guy over here, this is what's called a dendritic cell. And this is one that's also only located in tissues. So we're not going to find it in the blood. And it has the same job as the macrophage. So this function of antigen presentation as well as phagocytosis, that is the job of the dendritic cell. And notice that it has all these long kind of spike-like projections of its cytoplasm. If you've studied the nervous system, maybe you've learned about the dendrites of neurons. And so that's where it got its name from. Okay, it actually does have a dark nucleus, not really showing up in this particular picture, but when we draw them by hand, a lot of times I draw sort of looking like this is our dendritic cell. And they use these large, long projections of the cytoplasm to sort of crawl along the tissues. That's what they're using, those like arms to crawl around the tissues to look for pathogens to eat. Cool. All right, so those are all the leukocytes that differentiate from myeloid stem cells. Now we're going to talk about lymphocytes. So what does a lymphocyte look like in a blood smear? Well, here's one pictured here. Remember, who are the lymphocytes primarily? We're talking about T cells and B cells. And the truth is, if you see an inactive, and we actually have another word for that you'll hear, naive. So if we talk about a lymphocyte being naive, it means it's inactive, it's circulating, but it hasn't been called into action yet by the immune system. So it's just in circulation. We call it the naive T cell or the naive B cell. Okay, one thing you'll notice, it has a huge circular nucleus that takes up most of the cytoplasm. It's huge, right? Usually stained purple. Here's usually how I draw it out. Okay, and that's what you would be looking for in a blood smear. Don't worry about the different types of T and B cells and differentiating them morphologically. So just know that if you see this, we would call this a lymphocyte, a T cell or a B cell. I would like you to know that there are different types of T cells as well as different types of B cells, but I think we'll wait to talk more about that in a future video. This video is getting a little bit longer than I intended. Sorry about that. Here's the last lymphocyte I want to mention, and this one actually does look a little bit different than the T cells and the B cells. So this is a lymphocyte called a natural killer cell. Or the NK cell. So natural killer cells, they also have some granules in the cytoplasm. So once again, a really large circular purple nucleus is visible in the histology of this particular cell. But then you also see a collection of granules in the cytoplasm. So this is a cell that also can degranulate, like we see in basophils and mast cells and eosinophils. And these granules, there's really two types that they have. We call them perforins, and also, like we talked about with eosinophils, granzymes. So these are perforins and granzymes. These are, again, the toxic proteins that can be released and can facilitate the main function of NK cells, which would be destruction of foreign pathogens, although NK cells also destroy cancer cells. That's one of the little known facts about your immune system, is that, remember, its main purpose is to destroy pathogens, but it also keeps cancerous cells in your body in check. So, although I'm guessing very few of you know or have had somebody in close to you who's had cancer, maybe you have, I don't know. But the process of getting a cancer disease is actually fairly rare, but all of us have had cells in our body become cancerous at some point. Think about any time you've had a sunburn, you have initiated mutations in your cells that could have led to skin cancer, but what happens is your immune system will destroy any abnormal cells that are growing out of control in your body. So if you haven't developed clinical cancer, it means you have a really strong immune system, including a strong natural killer cell response to abnormal cells. And so what they do is they release the perforins and the granzymes, and that will trigger a response to the cancer cell, and tell the cancer cell to self-destruct. Okay, so I thought you might want to take a look at this picture and just test yourself and see if you can figure out which white blood cell or leukocyte are we looking at in this blood smear. So if you'd want, you could pause the recording and you could test yourself, and then we'll talk about the answer. Okay, so now let's talk about the answers. What white blood cells are we looking at in this blood smear? So what are all the red ones? Oh yeah, red blood cells. So you see a lot of the red blood cells. It's primarily what your blood is made out of, with an occasional white blood cell. And they actually are white, by the way. They're being stained by these histological dyes that, so we can see them. Okay, far left corner. Oops, sorry. Far left corner. This is a monocyte. Remember, monocytes are inactive, huge, huge purple nucleus, and very large cell. Okay, how about this one over here? Very large circular nucleus, but a smaller cell. Yes, that would be a lymphocyte, a T or B lymphocyte. Up here with a very multi-lobed nucleus with some pink and purple granules in the cytoplasm, neutrophil. Over here, another neutrophil. And then over here, purple bilobed nuclei with pink granules, eosinophil. And then chock full, tons of purple granules in the cytoplasm, a whole bunch. Those would be basophils. So that's a basophil here. Anyways, I hope that was helpful, and we'll see you next time.